Good morning everybody, tēnā koutou katoa. My name's Steve Wakelin. I'm based here in Otatahi, Christchurch with Scion Research, and I'll be talking to you today about the Tree Root Microbiome Programme, which is an innovation hub for the future of the forestry sector. So the Tree Root Microbiome Programme is an exciting new innovation space which is really set up for tomorrow's forests. So as I said before, my name's Steve Wakelin and I'm presenting on behalf of the Tree Root Microbiome Programme team. So the problem statement that we face is that in the ground today, growing across all of New Zealand, are planted forests worth around $60 billion. So the forestry sector is sitting on a massive asset base. And we all know that the sector is somewhat unique in that it takes a relatively long time to realise returns. So roughly 3 decades sit between the day that stock is planted, you go out in the field and you plant your stem, and then when you can go and collect your wood and get your return on your investment. This inevitably means that there's an extended exposure to risk, whether that's from pests or diseases, weather or climate, or even national shifts in policy, policy direction, or access to markets. Of any sector, forestry has arguably the most highest exposure to risk, but also the brightest future if we can get it right. I mean, there's been no greater need for forests and forest products, and this need is growing. Benjamin Franklin famously said, "Change is the only constant in life. One's ability to adapt to these changes will determine your success." Given the pace and breadth of change in the forestry sector, it's clear more than ever that we need to be investing in new ideas that will allow forests and forestry not only to survive, but to thrive into the future. If the sector is going to adapt to risks, capture opportunities, we must have invention of new ideas and space and opportunity to develop and explore new ways of forestry. The Tree Root Microbiome Programme is an innovation engine room. It's there to provide the conditions for invention to occur. But before we go into what the Tree Root Microbiome Programme will be doing, let's quickly touch on what is creativity and what is the creative process. So creativity and invention is all about connecting dots, connecting things in entirely new ways. It's what happens when you're tinkering away in the back shed, in your art studio, your writing room, or your other creative space. You might, in fact, deliberately go into that space just to escape from other distractions and allow creativity to happen. It's the very start of the pathway that leads to new outcomes, whether for the forestry sector or the IT sector, doesn't matter. It's the start of the process. And without a stream of new ideas, innovation eventually dries up. Sectors that don't innovate stagnate. They're not able and agile to change. Typically, the most transformative and impactful outcomes come from unexpected, unlikely, and disparate connections of ideas. A good example is some tinkering from a group of scientists at the radiophysics lab at CSIRO, Australia. They were investigating black hole astronomy and particularly methods for signal correction. Now, it's very, very hard to get clear signals coming from black holes that are millions of light years away into your recorder in a room in Canberra, Australia. There's a lot of signal interference and bouncing around and things to consider. They paired up with some groups that were looking at brand new methods for signal correction and married these with some new mathematics based around Fourier transformation. It's hard to imagine, looking back, how they put these ideas together, but looking into this problem, when they connected the disciplines around signal and error correction and Fourier transformed algorithms, in doing so, they inadvertently solved a huge issue that was emerging for the IT sector at the time. They essentially invented Wi-Fi. That is, distortion correction for wireless networking. This is now used in about 5 billion devices all around the world and came out of the Black Hole Astronomy Lab tinkering around working out how to do signal correction through deep space before the samples got to their laboratory. So creativity and invention require quite specific environmental conditions to occur. And there's a really good understanding around what these are. So let's just try and understand these for a moment. So let's ask the screen legend, John Cleese, maybe one of the most creative people that has been in the last couple of generations, his thoughts on creativity and what makes creativity. Since retiring from the comedy stage, John Cleese has often been called upon by corporations such as Google and others to share his knowledge around creativity and the conditions needed to foster it. He draws from his own experience in his creative life, but also on the research conducted by Professor of Psychology Don McKinnon and others that was conducted in the 60s, 70s and 80s. First of all, it's really, really important that creativity is not a talent. It's a way of operating. Creative people have acquired a facility that gets them to switch into a way of operating that allows to open the creative part of their mind, to enter their shed or their art room. Professor McKinnon describes this as the ability and time for the mind to play with ideas and explore them, not for any immediate practical purpose, but just for its own sake. To get your brain from the closed and open and creative state, there are some key conditions, and these conditions can be created. The first one is space. Space away from usual pressures and demands. Nothing dampens creativity like interruptions, distractions, and constantly having to do things. The second one is time, setting boundaries around the space, a beginning and an end, providing an oasis that separates you from the doing mode, the day-to-day mode, but rather invests in sitting with and exploring problems from different angles for longer periods. And the final condition is confidence. Nothing stops creativity more than fear of making a mistake. The very essence of play is being open to whatever happens. If you think any direction could be wrong, you can't be in a creative mind. So the Tree Root Microbiome Programme is a space that has been deliberately made with specific time and resource boundaries to support and foster creative thinking on the role of the microbiome in tree and forest fitness. It was created with a very clear view of the need for the forestry sector to think and develop a base of new ideas and innovation needed to progress and support the sector for the next 20, 30, 50 years' time. It is there as an innovation space, providing opportunity for the sector to be agile, to develop new opportunities for shifting forest phenotype, increasing fitness, controlling disease, and maybe even altering wood properties. In short, if we don't provide the space for entirely new areas of thinking, innovation will stagnate. Changes imposed on the sector will outstrip our ability to respond or change our toolbox of options. What we know already is that microbiomes play or profoundly alter the systems that they inhabit. We know this from the human microbiome model. Often these outcomes are completely unpredictable and very transformative. We have a very strong evidence base already from previous investment in the forestry sector, the growing confidence in forestry futures and healthy trees, healthy futures programs show that the same is true for microbiomes in trees and forests as they are in humans and other model systems. And globally there is massive transformation potential for harnessing the microbiome. The question is, what are the creative, innovation, transformational opportunities for the Tree Microbiome Programme? So the Tree Microbiome Programme is around connecting the dots in new ways. An example will be connecting our knowledge around the radiata pine genome and our resources associated with genetic variants and variants trials across New Zealand and Australia. We're going to connect these with advances in analytical microbiome communities, both in or on plant tissue. And the image above shows a portable handheld DNA sequencer that plugs in via USB into a laptop. We're going to connect our plant genome information and the genotypes with all the microbial populations in entirely new combinations in entirely new ways. We're going to explore the outcomes of these interactions on the plant phenotype at the Plant Accelerator Phenomics facility in Adelaide. Essentially, we're going to ask, can we breed new phenotypes into Pinus radiata via the microbiome? Can we use these combinations, these new dots, these new ideas, as ways of generating new tools for disease resistance, drought tolerance, climate adaptation? We are connecting the plants and the microbes in new ways, and we don't really know what to expect that the outcomes of this might be. We need to expect the unexpected here, and that is where the confidence part of creativity comes in, being open to whatever emerges from this work. Another example is connecting the hypothesis that Darwin put forward in the 1800s, that the root of the plant acts like a brain and has a role well beyond anchoring the plant and sucking up water and nutrition. We're going to test this by eavesdropping on the chemical conversation that's happening between the plant root, the microbiome and the metabolome. Furthermore, we're going to model our approach on the human gut-brain axis, which tells us that the gut microbiome is in reciprocal communication with the brain and work together to affect the overall fitness of the host. And finally, we're going to be doing all this at the New Zealand Biotron facility based at Lincoln University, where we can use laparoscopes to go down into the soil and actually understand in real time all of these interactions that are happening on the root microbiome soil surface, and start to really understanding all of the changes and all the signals and communications that's happening between the plant, the microbiome and the environment. And we aim to answer these questions. Are roots cognitive? Can we show a pine forest how to adapt to change in the future that it hasn't seen before in the past? And finally, we're going to take advances in ecological DNA analysis, connecting those with advances in environmental typing, providing smart apps and standard operating procedures to laboratories all around the world to take up and use and go out in the field and measure Pinus radiata, its genomics in the environment, and a whole range of settings that New Zealand will eventually move into with a range of climate change conditions. We aim to set Pinus radiata up as a global model pine microbiome system for everybody to use. By engaging the global scientific community with us on this process, we will empower the adoption of the model and the de facto standard for tree microbiome research globally. We just imagine how powerful it would be for New Zealand's forestry sector if we had labs internationally all focusing on Pinus radiata, growth and productivity, and all developing new tools for manipulation and assessment for New Zealand. It would have incredible value. So what will the forestry sector outcome be? Ideally something that we're not even prepared for yet. A discovery or discoveries that will enable entirely new and transformative opportunities for the sector. The outcome's going to be a sector that is more resilient and robust and agile to a changing future. And in short, the sun has never shone so strongly on the forestry sector. The demand for forests and forest products is growing rapidly. There's lots of opportunities for the sector to innovate, to change and to realise these. And the Tree Root Microbiome Programme is proud to be on part of this journey with you. That's the end of my presentation. I thank you all for your attention and look forward to questions at the end of the session.